Hello everyone, welcome to the ITLS Academy. Today we are going to discuss the topic that is your estimation of protein, or you can say that the protein separation by the Lorry's method. Okay, first of all, what is this protein R? So we all know that protein is a biomolecules. Protein is a biomolecules and macronutrients. Macronutrients means that it is required in a huge amount. Okay. And this proteins are what? Proteins is a long chain of amino acids. Simply, this is a protein. So, what we have to do? We have to um, separate the proteins by this Lorry's methods. Okay, so let's discuss the session. First of all, what is the aim? To estimate the amount of protein present in the given sample by Lorry's method. This is our aim. Now, principle. So, the Lorry's protein assay is a biochemical assay or assay for the determination of total level of protein in a solution. The methods combine the reaction of copper ion with peptide bond under alkaline condition with the oxidation of aromatic protein residue. So this is a principle. Continue. The Lorry's method is the best method. The Lorry's method is best used with protein concentration of 0.012 1 milligram per ml. Milligram per ml, and is based on the reaction of copper ion produced by oxidation of peptide bonds with fallen reagent. This reaction involves reduction of fallen reagent and oxidation of aromatic compound. Means that when we use the fallen reagent, then the fallen reagent is reduces and or fallen reagent is reduces and um, fallen reagent is reduces and the aromatic compound is oxidized. Oxidation of aromatic compounds. And that aromatic compounds are what? Mainly the tryptophan and tyrosine. Tryptophan and tyrosine. In addition, tyrosine and tryptophan residue of protein process the reduction of phosphopolymelate and phosphotungsten component of fallen phenol reagent compound of fallen phenol reagent to form a bluish color, to form a bluish color. The concentration of the residue is measured by the absorbance maximally 750 nanometer. Okay, now I am going to show you the reaction. Here the R group is present, double bond with O. Okay, in the presence of copper ion and OH negative. Now this N form a bond with copper ion, and here it is like that. This is N O, and here the O is attached. Okay, so this is your tyrosine, tryptophan, and cysteine. It give molybdenum and tungsten blue in the presence of fallen reagent. Okay, this is about the principle of this experiment, that is your protein estimation. Now come to the reagent, come to the next topic, that is your reagent preparation. Now we are going to discuss about the reagent preparation. So for reagent preparation, first of all, we have to prepare a reagent A. This reagent A is prepared by the two solution. The one is your sodium carbonate and zero point 0.1 normal sodium hydroxide. How we prepare it? For this, you have to visit our YouTube channel and see the solution preparation video. Now, what we have to do, first of all, we prepared the zero point 0.1 normal of sodium hydroxide in 100 ml. Then we add 2 gram of sodium carbonate into zero point 0.1 normal of sodium hydroxide solution. Simply what you have to do, you have to prepare the zero point 0.1 normal of sodium hydroxide in 100 ml. Okay, this N is your normality. Okay, then after that you have to add 2 gram of that sodium carbonate into it, because you have to prepare a 2%. This is how we prepare the reagent A. Next is your reagent B. So for the preparation of the reagent B, again we require two chemicals. The one is your copper sulfate and the second one is your potassium sodium tartrate. Potassium sodium tartrate. Okay, you have to prepare potassium sodium tartrate 1% and 0.5% copper sulfate. Now for this, what we have to do, first of all, we have to prepare a 1% of this solution, that is your potassium sodium tartrate. So what we do, we take 1 gram of this potassium sodium tartrate and dissolve into the 100 ml of the distilled water. Then we weigh 0.5 gram of copper sulfate and dissolve into it, because you have to prepare a 0.5%. This is how we prepare the reagent B. Reagent A is prepared, reagent B is prepared. Now we have to prepare a reagent C. Reagent C is prepared by the combination of A and B. A and B. And it is known as, and that reagent C is known as a alkaline copper sulfate. Alkaline copper alkaline copper sulfate. Now what we do, we take we have to prepare the solution C 50 ml. Now for this, what we have to do, we take 49 ml solution A and it will be dissolved into the 1 ml of solution B. Or you can say that we take 49 ml of solution A and 1 ml of the solution B and mix it. That is known as a reagent C. That is your alkaline copper sulfate. Then we have to prepare a third reagent, fourth reagent, that is your reagent D. For the preparation of the reagent D, we required fallen reagent. Fallen reagent. Okay, so we dilute the fallen reagent at a ratio of 1:1. Means that if we take a 1 ml of the fallen reagent, then we take a 1 ml of the distilled water, and it should be prepared according to your need. Now, next is your protein solution. So here we required a one protein, that is your BSA, which is known as a bovine serum albumin. Okay, bovine serum albumin. So first of all, we have to prepare the stock of BSA, and it is prepared by the dissolving of the 50 milligram of BSA into 50 ml of distilled water. Now with the help of the stock, we have to prepare a working solution. Okay, for the preparation of the working solution, what we do, we take a 10 ml of stock and 40 ml of the distilled water. Then we use this working solution in an experiment. Okay, what is the stock, what is working, that we are already discussed. You can see that video. Okay, now next is your procedure. After reagent preparation, next is your procedure. So for procedure, we required 7 test tube. 1, 2, 3, 4, 5, 6, and 7. Then in each test tube, we have to add a different concentration of the working solution, and it is start as a 0.2 ml of working in a first test tube, then 0.4 ml working in a second test tube, then 0.6 ml working in a third test tube, then 0.8 ml working in a fourth test tube, then 1 ml working in a fifth test tube. Now in a sixth test tube, you have to add 1 ml distilled water, and in a seventh test tube, you have to add 0.2 ml of your sample. Then you have to make up volume, make up volume 1 ml of each test tube by distilled water. Now with the help of the distilled water, we have to make up a volume of 1 ml of each test tube. Now in a first test tube, for making 1 ml of the volume, you have to add 0.8 ml distilled water, then 0.6 ml distilled water, then 0.4 ml distilled water, then 0.2 ml distilled water. In a fifth test tube, we are not going to add any amount of water because it is already 1 ml. Same with this. It is already 1 ml. Then in the last test tube, we have to add 0.8 ml of distilled water. Then after that, what we have to do, um, we have to add 5 ml reagent C into each test tube. Add 5 ml reagent C into each test tube. Okay, so 5 ml reagent C, 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 and 5 ml reagent C. Then after that, we have to incubate it at a room temperature for 10 minutes. Incubate it at room temperature for 10 minutes. Okay, after 10 minutes, you have to add 0.5 ml of reagent C into reagent D into each test tube. Add 0.5 ml reagent D into each test tube. Reagent D means that fallen reagent. Okay, so add here 0.5 ml fallen reagent, 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 0.5 ml fallen reagent. After that, you have to incubate it for a 30 minute at room temperature in a dark. Incubate it at room temperature for 30 minutes in a dark. Then after 30 minutes, we have to take a absorbance. Take the absorbance at 660 nanometer. After taking absorbance, we get a graph like this. And this graph is in between the absorbance and concentration of protein. Then we get what? We get the straight line, and that sample concentration are lie in between this. Just a minute. So here we calculate the value and get the result. So this is all about the protein estimation by Laurie's method. I hope you will understand. But if you have any query related to this, you are freely to ask me in a comment box. Or for a further detail, you can follow the ITLS Academy on different platforms, that is Instagram, Facebook, Twitter, YouTube, WhatsApp, and LinkedIn. And subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. You can also ask your query to the helpline number that are already mentioned in your slide, that is 7080833450, and visit our website, that is www.itlsacademy.com. Thank you for watching. Please like, share and subscribe our channel.